I'm Charles Bartlett from the University of Delaware, and I'm going to provide a brief overview of the regulatory importance of planthoppers. Planthoppers are the superfamily Fulguroidea within the order Hemiptera, suborder Acinonycha. Worldwide, there are 21 families and 12,500 species. North of Mexico, there are 12 families and about 915 species. As the name suggests, planthoppers are jumping insects that feed on plants at least as adults. They have piercing-sucking mouthparts that are comprised of multi-segmented beaks that arise from the underside of the head. While some planthoppers rest openly on plants, others are cryptic, resting in leaf axils or at the base of the plant. Adults may disperse on plants or on non-plants such as textiles, mail, or baggage. While planthoppers are mostly plant feeders, the nymphs of Achielidae and Durbidae are fungivorous. While planthoppers are usually said to be phloem feeders, that may be an oversimplification. Most planthoppers are host specialized, feeding on one or few host species. But unfortunately, the host range of many species is poorly understood. The plant host serves not only as a source of food, but as a focus for mating and reproduction, which is reinforced by the type of communication they have. Planthoppers have substrate-borne communication, which helps them identify planthopper species and recognize mates. Planthoppers have simple metamorphosis, which is to say the immatures resemble the adults, and they do not have a pupal stage. Sexual reproduction is the rule, and parthenogenesis, though it occurs, is extremely rare. Eggs may be inserted into the plant, for example in Delphasidae, where they may be adhered to the plant surface. There are 5 instars. Nymphs may occur openly on the plant, particularly covered with wax. They may be hidden on the plant, or nymphs may be subterranean. While planthoppers are mostly univoltine, particularly the larger species, some species, such as most delphasids, are multivoltine and continue to reproduce as long as food is available. In temperate regions, the overwintering stage is usually the egg, although in some cases it may be late instars or adults. Wing polymorphism is common in many groups of planthoppers. And in such planthoppers, they may have long-winged forms or macropters and short-winged forms or brachypters in the same population at the same time. Normally, for species that are like this, there is a genetic switch that occurs in the 3rd or 4th instar, such that if the populations are high or host plant quality is declining, most of the nymphs become dispersal form macropters. If host plant quality is good and the conditions are not crowded, they mostly become brachypters. Some species are long-distance migrants. In particular, the brown planthopper is known to migrate from China to Japan over the East China Sea, a distance of about 500 miles. About 150 planthopper species and 16 families are known to be pests on about 100 plant commodities. Key pest species include the brown planthopper, the small brown planthopper, and the whiteback planthopper, which has so far been excluded from the New World. In contrast, the corn planthopper is endemic to the New World, but now is found worldwide wherever corn is grown. In contrast, the sugarcane planthopper is an Old World species that has been introduced into the New World. It is native to Australia and the Indo-Malayan region, but has been introduced into Hawaii and the continental U.S. and is now found wherever sugarcane is grown. Damage from planthoppers is caused by direct feeding and oviposition, which can cause chlorosis or provide a place of entry for plant diseases. Also, planthoppers can cause what's called hopper burn, which is caused by the combination of toxic saliva and the plant response to that saliva. Hopper burn usually originates near the site of feeding, but may be extended progressively as planthoppers feed elsewhere on the plant, eventually killing the plant. But most importantly, planthoppers are vector of plant pathogens, with 36 planthopper species vector in 4 families, vector in 34 plant diseases. Most of the planthoppers that vector plant diseases are delphasids, which vector mostly plant viruses. 28 delphacid species vectored plant pathogens in 38 crops, including rice, corn, sugarcane, and cereal crops. In contrast, succeeds are mostly bacterial pathogen vectors, vector in lethal palm yellowing in over 30 species of palms. also succeeds vector pathogens in strawberry, sugar beets, and grape. Durbids and flattids are also plant pathogen vectors, although their role in plant pathogen transmission is still under investigation. 11 species and 6 families are inventive to the United States, and 2 additional species may be inventive, but their endemic status is still unclear. Most of the major crop pests have so far been excluded, except Perkinsiella sarcaricida, which is the sugarcane plant hopper, which was introduced in the 80s and is now widespread wherever sugarcane is found. Some pests have so far been excluded. This includes the 2 main rice hoppers, the brown plant hopper and the whiteback plant hopper, the small brown plant hopper, which is a disease vector in corn, rice, and cereal crops, the dubus bug, which is a pest of date palms in North Africa and the Middle East, and the succeeded plant hopper Hyalestes, which is a pest of grape and tomato. Some of these same species have been intercepted by USDA-APHIS at ports. This includes the brown plant hopper, which is a major rice pest, the taro plant hopper, which is now adventive to Hawaii and has been intercepted at continental ports, the torpedo bug, which is now adventive to Hawaii and California, and the grainy plant hopper, which is now adventive to Hawaii and often intercepted at continental U.S. ports. For identification of plant hoppers, adults are required, and immatures can usually be identified only the family and not beyond. Males are often required for species identification. Females sometimes can only be identified to genus or sometimes tribe. Identification tools are often limited by family, crop, or region and may be outdated or incomplete. Finally, access to primary literature or authoritatively determined diagnostic collections may be necessary to confirm species identification. Problems in developing identification tools for plant hoppers are that the systematics of plant hoppers are poorly understood. The basic research needed to create identification tools for many groups of plant hoppers has not been done. Also, many genera are in need of revision, and there are many new species that have yet to be described. Nevertheless, most pest species can be identified with currently existing tools and online identification guides. These are our photo credits, and my collaborators. I'd particularly like to thank the USDA, and are there any questions?
How important is species recognition for plant hopper identification at ports?
Well, in some cases, it's very important. For example, if you get a Sogatella, it makes a great deal, whether it's the endemic colophon or the exotic Persifera, since Persifera is an important rice pest and colophon is an endemic of no substantive importance. In other cases, if you recognize it to be a native taxon or a genus that's native, it may not matter as much. Or alternately, if you recognize that it's an exotic taxon, it may not matter as much exactly which species it is.
How likely are we supposed to intercept rice plant hoppers and small brown plant hoppers at U.S. ports?
It's very likely, I would say. We have already intercepted brown plant hoppers, and I would anticipate it's very highly likely we'll get, for example, the whiteback plant hopper, the small brown plant hopper. All of those species are very mobile species, and they are the sorts of things that we're likely to see. So it's important that we monitor for these species.
If a plant hopper species is not known to be a pest species or transmit plant pathogens, why should we be concerned about it being accidentally introduced into the U.S.?
Well, we should start by understanding that the vector biology of most species is not known. So just because it's not known to be to transmit plant diseases doesn't mean that it doesn't transmit plant diseases. I would say we're in our infancy in understanding which plant hoppers transmit disease and why. And there's also the law of unintended consequences when you get a species into an area where it lacks predators or other checks on its populations. It may have unexpected implications for what might happen from that introduction.